Yeah.
I'm sorry about that.
Yeah, that's fine. Yeah, it's great to be here and to have the opportunity to talk about my work. So the first, so I'm going to be talking, as Chris said, about glutamate and ketamine, and I've been working in this area for the last 15 years or so. But it's kind of developed from early interest in ketamine as a model of psychosis into more recent work around its use as an antidepressant. So ketamine came to my interest initially because of its action on the NMDA receptor. So the NMDA receptor dysfunction hypothesis of schizophrenia is an idea that's been around really since the middle of the 1980s. This is just a revision slide about NMDA receptors that I'm sure you all know already. Voltage and ligand gated ion channels that allow the passage of calcium ions with a magnesium ion block, and the intrachannel site that can bind ketamine or PCP and other compounds, which blocks the channel. So ketamine is an uncompetitive NMDA receptor antagonist that binds at this intrachannel site, blocks the passage of calcium ions into the cell, regardless of the depolarization state of the cell. And it's particularly interesting because of its effects. It was developed as an anesthetic agent, and it's called this term dissociative anesthetic because of its interesting effects on consciousness. And at sub-anesthetic doses, it can induce perceptual changes, including perceptual distortions and delusional-like ideas, and also has been suggested to cause effects that resemble the negative symptoms of schizophrenia. So this is one of the reasons why it was of particular interest as a model of psychosis. So people noted that there are similarities between ketamine, the effects of ketamine, and some of the symptoms, particularly in the early stages of schizophrenia. And this led to the suggestion that changes in glutamatergic neurotransmission might be present in people with schizophrenia, which led in part to the NMDA receptor dysfunction hypothesis of psychosis. So this idea, I think two of the pioneers of this work were Olney and Farber. They wrote a nice review in 1995 about their work. And they were looking at the effect of NMDA receptor antagonists in rats. And they showed that drugs like PCP and MK801 could lead to toxic changes in cortical brain regions in rats. And they could block this effect by using AMPA receptor antagonists. And they suggested that this might be that these toxic changes might be due to excessive glutamate release caused by ketamine, which was later confirmed in microdialysis studies. And they suggested also that as these drugs caused schizophrenia-like symptoms, that these kind of brain changes might also underlie some of the brain volume changes that are seen in schizophrenia. But the question is how an NMDA receptor antagonist might lead to increases in glutamatergic neurotransmission. And so it wasn't immediately obvious how this might happen. What they suggested was that NMDA receptors that are expressed on GABAergic interneurons might be more sensitive to the effects of ketamine than those on other neurons, and so you get a preferential effect of blocking these NMDA receptors, which reduces the inhibitory drive on glutamatergic projection neurons, and this leads to downstream elevation in glutamate levels. And they suggested that maybe something similar might be happening in schizophrenia, that there could be GABAergic interneuron dysfunction, and that this leads to the same kind of downstream glutamatergic elevation in schizophrenia. So this was a really kind of interesting model that they put forward, but at that time point there wasn't really any evidence that this actually did occur in schizophrenia. So there were no imaging data or anything. So this is one of the first areas that I was interested in looking into. So one of the first questions we looked at is whether there's any evidence of NMDA receptor dysfunction in schizophrenia and how it relates to symptoms. So the way we looked at this was with a SPECT tracer called 123I-CNS 1261. And this was a drug analog that binds to the PCP site in the NMDA receptor. And it's obviously radio labeled, and you can detect the gamma rays that are emitted by the decaying radioactive iodine and reconstruct it into a brain image like this, top right hand corner. I'm not sure if you can see my mouse. So we use this tracer to look at patients with schizophrenia and also healthy volunteers. We found that in medicated patients with schizophrenia looked quite similar to those to healthy controls, but drug patients with schizophrenia who are not currently taking any medication had reduced binding to this NMDA receptor site compared to healthy controls. This was work that we published in 2006, and it was interesting because this really overlapped onto findings from post-mortem data. This is from Amanda Law and Bill Deakin, published in 2001, and they looked at NMDA receptor mRNA in the dentate gyrus, and they found a very similar pattern to the one that we found, particularly that the left-sided reduction in the NMDA receptor mRNA in patients with schizophrenia compared to controls. So that was really we also looked at the correlation of NMDA receptor availability in these patients, and found that the patients who had the lower levels of NMDA receptor binding by this tracer had higher levels of negative symptoms. So this is some evidence that there might be some basis to the symptoms of schizophrenia from this NMDA receptor deficit. Obviously a very, very small study, but it wasn't no one went on and tried to replicate it. The tracer we were using was stopped being produced. Sadly, Lynn became ill and couldn't carry on working on any of this area anymore. And it wasn't until it's only just that we have managed to repeat this study using a PET tracer, and the data for that have just been submitted for publication, not yet published, but we found basically exactly the same thing in patients with unmedicated patients with schizophrenia, that they had lower levels of NMDA receptor binding compared to healthy controls. So we wait to see if this goes any further. The next question that I was particularly interested in was if there's any evidence for increased cortical glutamate in patients with schizophrenia, because if you recall, the Olney and Farber model suggested that there were NMDA receptor dysfunction, and this led to downstream elevations in cortical glutamate. So to look at this, I used proton magnetic resonance spectroscopy, which is a nice imaging method using a standard MRI scanner, and we can get a measure of brain metabolites using this, and these peaks correspond to different metabolites. This double peak in the middle here, which is labeled GLX, is equivalent to glutamate and glutamine, which are of interest in terms of neurotransmission. So other groups one group had already looked at this, the Beige, who published in 2002, and found that patients with first episode psychosis who are unmedicated had elevated glutamine, which is a precursor to the glutamate, in the left anterior cingulate. And for my PhD, I looked at this in people who are with so-called at-risk mental states, so at high risk of developing psychosis. They had sub-threshold schizophrenia-like symptoms, and found exactly the same thing, that there was increased anterior cingulate glutamine in this brain region. Subsequently, there have been a number of other studies looking at this, and it's interesting that from this meta-analysis in 2011, it looked like there was possibly evidence of increased levels of glutamine in frontal cortex in people with schizophrenia or first episode psychosis up to about the age of 30. But after that, the evidence decreases. So this suggests that there could be, if there are changes in this glutamate level in psychosis, it might happen early on in the illness. So this so far gives evidence that NMDA receptors may be altered in patients with schizophrenia, and also that perhaps in the early phase of psychosis, glutamine levels may be increased. But the next question that I was interested in was whether ketamine has these effects in humans. So does ketamine block NMDA receptors in humans? This was the first study that I was involved in. Again, part of my PhD, so a long time ago now. So this was, again, using the same spec tracer, CNS1261. And this was in healthy volunteers. And in healthy volunteers, we gave them either, we started the infusion of the tracer, and then at around 180 minutes, we started ketamine or saline as an intravenous infusion, and then this stopped at around the 4-hour point. It was a very long scan. And we looked at the difference in terms of binding potential between the scan time when with the ketamine involved or with the saline. And we looked at various brain regions using a standardized atlas. And we found that basically in every single brain region, there was greater reduction in volume of distribution with the ketamine infusion compared to saline. So there's a significant change in baseline for basically all regions. We also looked at the correlations in terms of changes in this ketamine binding to the NMDA receptor and schizophrenia-like symptoms. So we used this scale called the Brief Psychiatric Rating Scale, BPRS, which has positive, negative, manic, and anxiety scales. And we found that the correlations between these reductions in volume of distribution correlated just with negative symptoms. So the more kind of withdrawal type of symptoms that are seen in chronic schizophrenia seem to be the ones that were correlating most closely with this binding. So the next question, in order to look into this model of schizophrenia and ketamine, does ketamine drive cortical glutamate release in humans? So again, this had been some work around the mid-2000s. Laura Rowland did the first ever study of this using MR spectroscopy and gave an infusion of either saline or ketamine to healthy volunteers and looked at whether there was any changes in glutamate. And she found that there was an increase in glutamine, again, glutamine, not glutamate, immediately after the ketamine infusion. This is, on the right-hand side is a figure from Peter Mogadam's study in rats, which is using microdialysis. And this shows the pattern of neurotransmitter change following a ketamine injection into rats. And interestingly, in rodents, there's the immediate effect is on dopamine, and then there's a later phase effect on glutamate that happens around 60 to 80 minutes after the ketamine injection. So this kind of early effect from that was seen by Laura Rowland doesn't exactly match this study in rodents. So there have been some, and looking at changes in glutamate in rodents, this seems to be held up. The changes in glutamate happen later on to the kind of immediate changes in dopamine and possibly in glutamine. So this was a study where rats were treated with ketamine and had either in vivo MRS or ex vivo metabolite measurement. And again, after 6 days of treatment with ketamine, there was a significant increase in glutamate concentration measured by MRS. Not a significant change in glutamine levels though. And looking at the ex vivo data, the ketamine and the glutamate correlated. The glutamate measured by either ex vivo or in vivo methods correlated very closely. So this is quite reassuring for the MRS methodology. So I did a similar study in healthy volunteers looking at the effects of ketamine, but looking at a later time point than was looked at by Laura Rowland. And so this was around 40 minutes post-infusion. And we found an increase in glutamate, but not in glutamine. So this is of relevance. And we also did a study on looking at different doses of ketamine, and found that having higher dose ketamine caused much more of these kind of schizophrenia-like symptoms, including cognitive disorganization, delusional thinking, perceptual distortions. We found that the post-ketamine glutamate levels in the anterior cingulate were significantly related to positive symptoms of psychosis, so sort of the perceptual distortion type effects. Okay, so moving on with more of the discussion about ketamine and brain chemistry with respect to psychosis. So one question that I had towards the end of the 2000s was whether it's possible to use to look at brain chemistry to predict response to antipsychotic agents. So this was a paper I published in 2010, which was looking at the biological basis of antipsychotic response in schizophrenia, and suggesting that there could be different subtypes of schizophrenia because all of the antipsychotics that are currently available actually block the dopamine receptors, dopamine D2 receptors. But there's obviously this growing evidence that the glutamate system might be involved. And what I was suggesting was that there might be subtypes of patients with schizophrenia, or that some of them might have more of a dopaminergic basis to their illness, and others might have more of a glutamatergic basis. And those with a glutamatergic basis might be less likely to respond to conventional antipsychotic drugs. So we looked at this using MRS again in patients with first episode psychosis. The first study we looked at basically just patients who'd been within the first episode teams for around a year, and looked at those who are in remission or not in remission at the 1 year point. They would all have had antipsychotic treatment, which would have been dopaminergic antipsychotic treatment. We found that the patients who hadn't gone into remission tended to have higher levels of glutamate in the anterior cingulate. And furthermore, we found that this glutamate levels in this brain region had a negative correlation with global functioning. So the higher the levels of glutamate, the lower the levels of lower levels of global functioning, and also the higher the levels of glutamate in this brain region, the higher the levels of negative symptoms. So this really kind of fit very nicely with the hypothesis. Subsequently, we did some more work, and Oliver Howe's group as well did some work on this using MR spectroscopy and 18F dopa PET to look at the dopaminergic system. So Arsema, who did this study, looked at patients with schizophrenia, and looked at the first of all patients who are controls, then patients who are responders to antipsychotic drugs, and then patients who are non-responders to antipsychotic drugs, and again found the same pattern, that the non-responders tended to have higher levels of glutamate in the anterior cingulate and also in the left thalamus, But interestingly, it was the responders who had higher levels of F-DOPA, suggesting that the responders to the antipsychotic drugs were people who had more aberrant dopaminergic transmission. Again, fitting quite nicely with this model, and again replicated by someone else in Oliver House's group. This was in chronic patients with chronic schizophrenia, finding that treatment-resistant patients tended to have elevated glutamate in anterior cingulate cortex. However, this sounds all very exciting, but we looked. This was taken into a further study, looking at whether it was possible to use this to identify patients who were kind of poor responders to antipsychotic drugs. This is the Strata study, and using this for looking at glutamate and F-DOPA in these patients. This was a multi-center study, and there was a slight increase in anterior cingulate glutamate after correction for age and various other covariates. But as you can see, there's a huge overlap between responders and non-responders. So this doesn't really give very good confidence for taking this forward as a biomarker. Again, in striatum, there was no difference between the two, and in terms of F-DOPA uptake, there was no significant difference. It looked like there was a trend towards those non-responders again having slightly higher, if anything, F-DOPA, but there was no significant difference. So that's where we are now, and it's a bit disappointing, but I think it's still interesting that there could be that there could be differences in terms of neurotransmission and in terms of glutamatergic neurotransmission in schizophrenia, but I think more work needs to be done. So I'm just going to talk a tiny bit more about other glutamate methods that I've been using more recently. Well, we were obviously the MRS that I was using up to that point was just taking a measure from a predefined brain region and looking at the levels of glutamate in that region. And I became interested in the idea of whether it was possible to instead of just taking a static measure, to look at dynamic changes in glutamate. So this is work that various people have been doing over the years. One of the pioneers of it is Paul Mullins, who's based at Bangor University, and I've been collaborating with him on some of this. But this is work that we did at the Institute of Psychiatry, and we were looking at changes in glutamate using cognitive measures. So this is the two-back task, which I'm sure many of you will be familiar with, and looked at the changes in the differences in brain glutamate during this task. So the white area is zero back, and then the blue area here is two back, and this is the first. And these are divided up into time epochs. So this is basically over approximately a 45-second time period. So these are blocked over this time. And you can see that in healthy controls, over the zero back, glutamate levels tend to reduce. There seems to be a jump up between zero back and two back, and then during two back it might reduce again. In schizophrenia patients seem to be a completely different pattern, where during zero back glutamate levels increase over the time, and then during two back they may be slightly higher and then maybe decrease off over two back. The bipolar patients seem to have a similar pattern to the healthy controls, although the change there is slightly less. We basically found that there was a significant change between three back, between the end of the zero back and the beginning of the two back. But and so we have also we took this forward and did some further work using functional MRS. This is the most recent work, which has not yet been published. It's submitted for publication. This was work in collaboration with one of the professors at Imperial College, who sadly died last year, Jacqueline de Belleroche. She's very interested in zinc transporter 3 genotype, which found to be altered in post-mortem brains of patients with schizophrenia, and also she'd done some work on a cohort of patients with schizophrenia, and found that patients with schizophrenia tended to have were more likely to have the uncommon variant of the zinc transporter 3 allele. So we did a small study in patients with schizophrenia and also bipolar patients, looking at the genotype for this gene, and looking at the 0 back versus 2 back paradigm. And so this is using standard fMRI. See, looking at the bold activation, you can see that this is the light grey here is healthy volunteers. People with the common allele for this for the zinc transporter 3 gene tended to have more activation during 2 back over 0 back than those with the rare allele. Bipolar patients and schizophrenia patients showed the same pattern, but with a different level of activation. And then looking at the changes in glutamate using this, looking at this genotype, we found that in patients with schizophrenia with the common allele, that they tended to have greater activation during 2 back versus 0 back. So they showed the sort of standard pattern that you'd expect to see with healthy volunteers, whereas those patients with the rare allele showed the opposite pattern. So they had less glutamate during 2 back compared to 0 back. Good. Okay, so that's the first part of the talk about psychosis and glutamate. So there's growing evidence for NMDA receptor deficits in schizophrenia. Glutamatergic abnormalities might be more associated with negative symptoms. It's possible that increased medial prefrontal glutamate release occurs in the early phase of psychotic illness, and possibly that non-responders to conventional antipsychotics might have a glutamatergic basis to their illness. More recently, I've shown that genetic changes associated with psychosis can affect glutamatergic neurotransmission, and we're still an open question. Could neuroimaging be used to predict who will respond to different types of antipsychotic drug in the future? So so I'm going to go on to the next part now, which is talking about ketamine as an antidepressant, and this has emerged over the last 10 years, really, as doing a great deal of interest in this. So the first randomized controlled trial of ketamine as an antidepressant was in 2006 by Zarate, and he showed an immediate effect of ketamine on patients with treatment-resistant depression. It was a real breakthrough type study, because up to that point it was thought that it was necessary to treat depression for a minimum of 2 weeks before you could see any kind of improvement in depressive symptoms. The conventional wisdom when I started psychiatry is you have to treat someone for 4 to 6 weeks with an antidepressant before you can say whether or not it's worked. Whereas ketamine was showing an immediate reduction in depressive symptoms, and in patients with treatment-resistant depression, so patients who hadn't responded to any other antidepressants in the past. So the antidepressant effects seem to be maintained for around 7 days, for a long time after the immediate effects of ketamine had worn off. Similar effects seen in people with bipolar depression, and this is the standard dose that was given, 0.5 milligram per kilogram over 40 minutes. So this is some data from Zarate's papers, looking at the changes in depression scores. So this is over time, up to 7 days. You can see that with the ketamine, there was a significant reduction in depression scores for this. This is, again, depression scores in people with bipolar affective disorder, again showing that ketamine had a very dramatic effect on depressive symptoms in these patients who are extremely hard to treat with conventional antidepressants. Also a significant effect on suicidal thoughts as well, which would be distinct from its antidepressant effect. So that was of great interest. Another study here that also showed the same effect on suicidal thoughts from ketamine. So this is a fairly recent meta-analysis of the response to ketamine over time. And it includes a number of different studies, but you can see it's probably very difficult to see on the screen, but this shows that there was a significant effect of ketamine causing at least a 50% reduction in depressive symptoms that was seen from at least 40 minutes after the initial injection of ketamine. It's still there 2 hours, 4 hours, 1 day later, 2 days later, 3 to 5 days later. At day 7, there's still a significant effect of favouring ketamine, but by day 10, this effect is no longer significant in this meta-analysis. Same meta-analysis, but looking at remission. So this is patients in whom, once following the ketamine, that they would be identified as not actually being depressed. And this was there's a significant effect of ketamine on remission at 80 minutes that was maintained out to day 3 to 5. But by day 7, the effect was starting to be lost. So this gives you an idea of the kind of time pattern of ketamine. So there's some questions around clinical use of ketamine at the moment. This obviously requires regular dosing. Long-term risks are not really known, although there have been now studies of intranasal ketamine, which I'm going to talk about in a bit. So this is a picture. The central picture is a picture of a recovery suite in Oxford, where Rupert McShane was one of the first pioneers to give ketamine for depression. But recently, the Janssen has developed an intranasal ketamine spray, which has got through phase 3 trial and has been approved by the MHRA for treatment of treatment-resistant depression. So it's possible that this could become used more widely, but at present its use is fairly limited because of some difficulties around the level of dissociative symptoms that are caused. So this slide basically just gives an overview of the fact that ketamine comes as 2 enantiomers, the R and the S enantiomer, which have different potencies at the NMDA receptor. So the S enantiomer has a 3 to 4-fold higher affinity for the NMDA receptor than R-ketamine. And it was for this reason that Janssen chose S-ketamine to be the active component in their intranasal spray. This is their blurb about how the spray is administered. They were trying to compare the intranasal ketamine to get the same kind of PK profile as intravenous S-ketamine, and they found that the 84 milligram nasal S-ketamine gave a slightly higher level of plasma concentration than 0.2 milligrams per kilogram of S-ketamine, which is what they considered to be the antidepressant dose of the in its IV form. So this was how they chose the dose. They did a study in which S-ketamine was given to patients with depression alongside an oral antidepressant drug. They reduced the frequency of the S-ketamine administration, and they continued it for either every 2 weeks or once weekly, depending on patient response. And this was, they compared S-ketamine to placebo. And they found that by day 28, there was a significantly better response rate in those patients on S-ketamine compared to those just on antidepressant. So this is in terms of improvement, and this is in terms of remission. So you're getting around a 50% remission in the ketamine-treated patients at day 28. So this is quite good evidence to support this treatment. They then did a study to look at maintenance and switching people after people were maintained for 16 weeks and in remission. They were switched either back to placebo or to carry on S-ketamine. And interestingly, there was so this red line is the people that stayed on S-ketamine. You can see there was a divergence of the curves. But the interesting thing is that even though patients remained on just stayed on oral antidepressant, a good proportion of them, around 40%, remained well after switching off S-ketamine. So this is quite good evidence for how it might be used as a long-term treatment. But that's the kind of a quick overview of the clinical sides of it. But the interesting side from neuroscience point of view is what the underlying mechanism might be of this antidepressant effects of ketamine. So this is a diagram from a nice review that was published in 2015 about the effects of ketamine on depression. So as we talked about earlier, ketamine leads to increases in glutamate at the synapse, activating AMPA receptors. And the downstream effects go through these second messenger systems. Hypothesized that one of the main pathways is through mTOR, and then leading to downstream effects on synaptogenesis. And it's the synaptogenesis and increasing in dendritic spine formation that's thought to underlie the antidepressant effects. So why do they think mTOR is important? So this was a study looking at rodents in terms of and forced swim test. So this is blue is saline, red is ketamine. So ketamine led to reductions in immobility, but when rapamycin was given as well, there were no changes. There were no differences between ketamine and saline. So this suggests that the mTOR is important for the antidepressant effects of ketamine. Secondly, ketamine effects, as hypothesized here, involve glutamate release, as we've already said. So we know that ketamine given to rodents can lead to increases in glutamate levels using microdialysis measures. But is this glutamate release important for the antidepressant effects? Well, it looks like it probably is. If you, again, using the forced swim task, rodents given ketamine showed a reduction in immobility time, but with increasing doses of NBQX, which blocks AMPA receptors, this effect of ketamine was lost. So it suggests that that glutamate release is quite important for this effect. And the last thing is the effect on dendritic spine formation, which is at this point here, synaptogenesis. So again, in rodents, given ketamine showed that showed that the ketamine led to an increase in dendritic spine formation, which is significantly higher in the ketamine-treated mice than in the saline treated. So that's all the kind of preclinical work, but how does this translate into patients with depression? So I think it's important to have a look at what we know about depression first of all. in patients with depression. So one of the areas that's been really highlighted as being important in major depression is the subgenual anterior cingulate cortex. So this region is smaller in volume in patients with depression, and also patients with bipolar affective disorder who are not treated with lithium. When they're treated with lithium, that can increase to same sizes. It also appears to be overactive in patients with depression, and it's suggested that this might be one of the key areas that's important in depressive symptoms. Let me go back, because in patients with deep brain stimulating this area, they become their depressive symptoms reduce, and the overactivity in this area is normalized. So we also know that in this brain region there are reductions in brain glutamate in patients with depression. So it's possible that increases in glutamate might be important for the antidepressant effects. And lastly, there's seems to be increases in some of the functional connectivity in patients with depression to the default mode network. So this is the idea behind this, is that that patients with depression might spend more time thinking about, ruminating on negative thoughts and so on, and it's possible that kind of breaking down this connectivity, this level of connectivity, might be a way that ketamine could have antidepressant effects. So these are potential mechanisms for ketamine: either modulation of cerebral blood flow and metabolism, increasing median prefrontal glutamate activity, or reducing functional connectivity in the subgenual anterior cingulate. So these are a number of studies that we've done to look at this. Firstly, does ketamine increase resting state bold response? So this is a study from Bill Deakin, in healthy volunteers, where ketamine was given, and interestingly, ketamine led to increases in brain activation in most of the brain, but decreases in subgenual cingulate cortex activation, bold response. And we've repeated this in 2015 and found the same thing. So this is one mechanism that might be involved in the antidepressant effects of ketamine. Secondly, we've already talked about brain glutamate, and I showed that ketamine causes increases in glutamate. So this could be another way in which ketamine has its antidepressant effects. Again, this has been done in another group. Milak and colleagues showed that ketamine led to increases in GABA and glutamate in anterior cingulate cortex. But interestingly, they didn't find that these increases in glutamate correlated with the antidepressant effects. Only the levels of norketamine at 90 minutes correlated with the outcome. So that shows that ketamine had an effect on brain glutamate, but maybe it wasn't driving the antidepressant effect. Lastly, functional connectivity. There's been a couple of studies in healthy volunteers looking at connectivity in patients with schizophrenia. So not in patients, in healthy volunteers on ketamine, and showing that there was reductions in connectivity between the subgenual cingulate and dorsal nexus with ketamine. And we did a similar study looking at healthy volunteers on ketamine, and showed again a reduction in connectivity with this subgenual cingulate cortex region post ketamine infusion. So again, possibly a mechanism by which ketamine could have its antidepressant effects. Obviously, we've shown all of these things, but these aren't in patients with depression, so still need to do some more work to find out if these are really important for the mechanism of the antidepressant effects of ketamine. So that's the main basis of the talk. Last part of the talk, I just wanted to talk about whether ketamine actually does work through the NMDA receptor and through NMDA receptor blockade, because this has become a bit of a contentious issue. So going back to my first study from my PhD, looking at ketamine in healthy volunteers, just to highlight the effects that the ketamine I gave had, a lot of these symptoms that were induced, or a lot of the effects that were induced by the ketamine were positive type effects, positive psychosis-like effects, but there are also some manic type symptoms as well. So you can see that interestingly, the negative symptoms here were the ones that came out as being significantly associated. But if you look at all the positive, negative, and anxiety symptoms, they're all negatively correlated, although some of them non-significantly. Whereas the manic symptoms go in completely the opposite direction, non-significant, but it suggests that the manic symptoms are occurring, but maybe not through NMDA receptor blockade. This is a bit of a kind of bit of a stretch of this data, but the reason I looked at this is because there's been other work on whether NMDA receptor antagonism is really required. This is a paper from Panos Zanos, which was published a couple of years back now, which has really kind of questioned whether NMDA receptor blockade was important for the antidepressant effects of ketamine. And his group looked at metabolites of ketamine, amongst other things, and as well as NMDA receptor antagonists, and looked at whether these were really which of them had the greatest effect on the forced swim task. So I'll just go through each of these figures in turn. So first of all, here looking at MK801. So this is pure NMDA receptor blockade. This had a significant effect at 2 doses on immobility time compared to saline. So that suggests that, and also ketamine did as well. 24 hours later, though, the MK801 treated rats didn't show any maintained effect on immobility, whereas the ketamine treated rats did. So there's something different that's happening here with the ketamine to the MK801. Secondly, looking at metabolites, hydroxynorketamine. So looking at the S enantiomer of this metabolite, there were no effects on forced swim. But looking at the R enantiomer, 2R, 6R hydroxynorketamine, there was a dose effect in rats on forced swim task. Then going on to 1 hour after treatment, there was ketamine, hydroxynorketamine, and high-dose hydroxynorketamine. All of them could be blocked by NBQX, so suggesting that there was some effect on AMPA receptors here. And it was also maintained at 24 hours. So basically, this suggests that this R enantiomer might be more important as an antidepressant for the antidepressant effects of ketamine. And if you recall, the R enantiomer of ketamine has a much lower affinity for the NMDA receptor. So this suggests that maybe something else is going on in terms of the mechanism of action of ketamine. Very lastly, I wanted to also highlight this recent paper that looked at ketamine, blocking the antidepressant effects of ketamine using naltrexone, which is mu opioid antagonist. And one case series of 7 patients, it was shown that treating with naltrexone led to a reduction or an inhibition of the antidepressant effects of ketamine. But a subsequent study in alcoholic patients who are being treated with naltrexone suggested that ketamine may still have an effect in these patients. So it's jury's still out about this, but one of my PhD students is currently looking into whether we can block the effects of ketamine using naltrexone, and this is an imaging study, so looking at the effects of ketamine on brain glutamate and whether naltrexone has an effect on this. So this is still work in progress. So in summary, there's studies to date suggesting that ketamine is an effective antidepressant. The mechanism of action is likely to require modulation of brain metabolism and connectivity. The evidence for glutamatergic modulation as the key component is less strong at the moment. And its evidence suggests that molecular mechanisms might occur through AMPA receptor agonism rather than NMDA receptor antagonism. And we're currently working in my lab to investigate the effects of the mu opioid receptor blockade on ketamine-induced changes in glutamate. So lastly, just like to thank people that I've worked with in the past, and thank you all for your attention.
Thank you very much, James. That was fascinating stuff. And we've got lots of questions in the chat.
Okay.
I don't know if anyone wants to ask any questions with a mic, and if not, I'll read some of these from the chat.
Hi, Chris. Since I have to go like in about 5 to 10 minutes, is it possible to get a question in before that?
No, go ahead.
Thank you. Thank you for the talk. That was really, really good. I'm especially interested in the effects of ketamine and depression, actually. I'm wondering if I'm kind of taking this too far, just as to entertain the idea, considering the fact that SSRIs have been the first line of treatment in depression. Is there any connection with ketamine and the serotonergic system, or maybe like no connection at all, to make it sound as if ketamine works in a completely different way for this?
Yeah, I think it's I think there are interactions between serotonergic and glutamatergic neurotransmission. I think given that ketamine is has such a unique kind of characteristic in terms of its antidepressant profile, that it's unlikely that the two mechanisms are the same. But yeah, I think it's a good question. But yeah, I think ketamine is probably the interesting parts of ketamine's action probably work through different ways from SSRIs.
Such an interesting thing in and of itself, I guess. Thank you very, very much. That was amazing. Okay, I have to go.
Great.
Thank you.
I'll ask a couple of questions from the chat going backwards. Di asks, NBQX lowers activity in its own right. Will reduce activity in forced swim on its own?
Okay, that's interesting. Let me get that slide up. Trying to remember which slide that was now.
It was quite a late one.
Oh, it's one. Yeah, that was on the Zanos paper. Yeah, this one. Yeah, NBQX. So NBQX didn't have an effect on forced swim there. It blocked the effects of ketamine on forced swim. Is that right?
Di, where are you still there? You're muted.
I must admit,
I'm unmuted. I'm having difficulty in reading that the graph, but I mean there are a couple of comments within that. The forced swim test isn't a test of antidepressant activity at all. It was introduced by Porsold simply as a measure of something that was reactive to antidepressants. So I wouldn't go too far in interpreting forced swim test data in terms of therapeutic effects or mechanisms in humans at all. But the NBQX story, the NBQX is a muscle relaxant. And so you'd expect NBQX to have...
You've muted yourself.
Oh, you muted it.
And again.
Oh, again.
I'm back again. NBQX is muscle relaxant, so you'd expect it to have major effect on any measure which requires locomotor activity through its action, probably in spinal cord. What I would say is that I wouldn't treat ketamine NBQX interactions as being very specific at all to do with central effects in the cortex of ketamine. Sure.
Yeah, I think that's really interesting. Thank you very much for giving that feedback. All the preclinical stuff is really kind of I put it in there as interest, but it's not really my field. So it's great to have your expertise on that. I think that from my understanding, the data from this have been taken forward, and there are trials currently underway looking at the 2R6R hydroxynorketamine as an antidepressant in clinical trials. So the human data will be much more interesting, I guess.
That's certainly true.
Okay, I'll just ask this last question. I'm sorry, I don't think we'll have time to go through all of the questions, but I will save the chat, and perhaps we can spark up some additional conversations afterwards. So Richard asks, the GWAS studies in schizophrenia indicate a very wide heterogeneity of etiology. The findings of Egerton are not surprising. Do these GWAS studies help future glutamate investigations?
The GWAS studies help. I don't know. I think, I think I'll try and find the Egerton study. I think it's very difficult. I think schizophrenia research can lead to problems. It often seems that you go down one path and it's all looking very promising, and then something happens so as to disrupt what seemed to be a very nice hypothesis. So I'm not really sure as to whether the glutamate theory and the idea of whether we could look at subtypes of schizophrenia is killed off entirely by this, but it's I think it's less promising. As you say, it's not surprising given the great heterogeneity of the illness and the fact that GWAS studies are hypothesizing multiple genes of small effect. I'm not really sure if we can use that information particularly. I don't know if the person who asked the question would like to say anything, if they've got any ideas about how it might be used.
Hi there, it's Richard Whale. I was just thinking about the sort of inflammatory indications that come out from the GWAS stuff. So the HLA gene is one of the biggest predictors of association, and I think there's a lot of synaptogenesis type stuff that's coming out, and some glutamate associated loci. But it's a complete minefield, which just reflects my idea about schizophrenia, really, tweeting it. So I just wonder whether it helped you in any way.
Not really, no.
I think I'm happy for people to hang out and chat further if there are people who want to chat to James, if James has the time. I have no reason to end this meeting now, but it is 2:00, so by all means, if people need to leave, and I will thank you again, James, for an absolutely gripping talk. Thank you.
Thanks very much.
James, can I ask you something quickly?
Hi, Mara.
It's about, in the first part of your study, how reliable is the separation between the glutamate and the glutamine in terms of certification?
It's a good question. We looked at this in the IP, and I know some people report that the glutamate and glutamine, you can't really distinguish them at less than 7 Tesla, but the the fitting seemed to be much better for glutamate than for the combined measure. So I think it's probably good, but the measure of glutamine is a bit more tricky, and we could only distinguish glutamine in around 60% of the patients. So
I wonder maybe if your results in responders and non-responders might improve if you had.
Oh, this one, yeah, the Strata study. So yeah, interestingly, the data at IOP were quite good, but the other groups' data were not so good. So I don't know, but we tried to standardize things as far as possible, but MRS is very, very hard to get. So I think need a bit of magic to get it working.
Yeah. Yeah, sure. And also the ability of the person who acquires the data to shim properly and all of that will have a massive impact.
How small a brain region can you do MRS in?
Well, you can go down to the standard regions that we're using are at 2 by 2 by 2 centimeters, so 20 by 20 by 20 millimeters. So that's a kind of reasonable size. You can go a tiny bit smaller than that. It's 3T, but you start to lose data. If you're not interested in glutamate, if you just want to look at N-acetyl aspartate, you can look at much smaller regions. You can do things like CSI, which looks at lots of multiple regions at the same time. You can get some kind of image of N-acetyl aspartate levels. Another method that you can use that has much better spatial resolution, but you need a higher field scanner for, is Glucest. I don't know if you've heard of that, but this is imaging of glutamate. I was wanting to do that on the new 7 Tesla in London, but I didn't get any money for that, unfortunately. But it's a nice method where you can get very high, basically within 1 millimeter type resolution, or 1 3 millimeter resolution. For
it's
measures
an imaging technique rather than spectroscopy.
Yeah, exactly. But it's only a single slice, as I understand it, at the moment. Is that right, Mara?
Yeah, I've never done it myself, but I probably yes. Still, it's a sort of technique between magnetization transfer and spectroscopy, a combination of the two.
Right. Yeah.
It's fascinating, but difficult to do.
Yeah. Great. Any more questions?
I guess I'll ask one more question. The when you were looking at your functional MRS thing with the NBAC task, you showed a plot of these responses, but was that from the whole brain, or was that from a specific region or some aggregate of regions?
Yeah, this was from the anterior cingulate. So it was not exactly the same anterior cingulate region that we looked at for other. It was a bit more dorsal, but yeah, it was. We chose the region based, based on fMRI data. So this was one of the regions that came up as being significant, as showing a significant signal on the NBAC, and so we chose that region to look at for the MRS, for the functional MRS as well.
I see. Cool.
Yeah.
Well, thank you very much. That was a really fascinating talk, and sparked a lot of interest. I will send you the text for the questions, and at least one person suggested that you should meet with them. That was Andrew Penn.
Oh yeah.
So
brilliant.
Yeah, I think I think you'll get lots of good feedback, I think.
That's great. Well, as I say, people are quite welcome to email me any questions they have, or if they want to meet up or whatever. Be great. Thank you very much. And I'm hoping to come along to the other talks as well. I've switched my clinical days, so I'm now able to come to these meetings. So good.
That's
great. Thanks very much. Okay. Bye-bye.
Thank you, James, and thank you, everybody.
Yep. Bye.